Cancer invasion. Healthy cells. Healthy epithelial cells relate to one another and to the extracellular matrix through contact inhibition. Contact inhibition is the mechanism by which a cell maintains terminal differentiation, regulates growth, and prevents cellular remodeling as a way of avoiding cancerous transformation. Tight intercellular junctions are maintained by adhesion proteins. This connection to neighbouring cells maintains contact inhibition. In healthy tissue, cells are attached to the extracellular matrix, which is comprised of the basement membrane and the interstitial matrix. Contact inhibition occurs between the cell and the extracellular matrix when cellular receptors such as integrin interact with components such as laminins in the basement membrane, but things can go wrong. Step 1. Altered cell-to-cell interactions. The first step in cancer cell invasion is the loss of contact inhibition between the neighbouring cells. This occurs when a mutation causes a cell to downregulate E-cadherin. This causes the cell to dissociate from its neighbours and start to look a little different. Step 2. Matrix dissolution. The second step in cancer cell invasion is matrix dissolution. In the case of a cancer cell, it either secretes or induces neighbouring cells to secrete proteases. For example, matrix metalloproteinases, heparinases, and serine proteases. Cancer cells may upregulate the expression of these proteases as they are capable of degrading proteins in the extracellular matrix, which then allows cancer cells to pass through it. Step 3. Altered cell-to-matrix interactions. The next step in cancer cell invasion is the loss of contact inhibition by the extracellular matrix. Cancer cells alter their integrin expression, changing the way that they interact with laminins in the extracellular matrix. Disrupting this interaction dysregulates the shape, orientation, and movement of cells, which promotes cancer cell invasion and metastasis. Step 4. Migration. To complete the invasion process, a tumour cell must alter its cytoskeleton so that it can move through the extracellular matrix. One way that tumour cells do this is by moving the leading edge of the cell to attach to the extracellular matrix, then detach at the trailing end and contract the cytoskeleton to move forward, similar to the way a caterpillar crawls. This movement is directed by autocrine pro-migratory factors as well as cytokines and cleavage products. This results in the cancer cell moving through the basement membrane where it can wreak more havoc. We have just gone over the process of how cancer begins to move through and destroy the tissue around it. Cancer is a nasty disease that affects millions of people worldwide, and it is important to understand how cancer develops and works in the human body so that one day we will be able to develop the most effective treatment. This video was brought to you by the students in the Cancer Pathology unit at the University of Western Australia.